Hello again, lecture 19. Um, as you can probably tell, I'm recording this right after lecture 18, so same glass of milk. Anyway, picture on your screen, rat astroglia, astrocytes, showing you both their processes and their nuclei. A little Christmassy perhaps, but obviously a beautiful picture. There are some really cool pictures in neuroscience. Today's news from the world of neuroscience. This is about a year old now. I just checked into it. As far as I can tell, this is the latest update. So this update, you'll see the rest of the story in a minute. This is a chap in Russia who has had lifelong crippling body issues, but whose brain and mental capacity is completely intact. He had volunteered to be the first candidate for a head transplant. Now you can imagine transplanting organs is bad enough. Transplanting a head, as well as getting the vascularization up to speed. So we saw in last lecture's kind of news from the world of neuroscience, there have been some really big advances in keeping cells alive and revascularizing and perfusing. So I might believe, in fact, you could take a human head and reattach the blood vessels. You may be even able to restart the heart of the donor body. Obviously, that'd be essential for further life. Reconnecting the spinal cord, that's the challenge. This is actually possible in some lower organisms. So if you cut through the spinal cord of some species of frog, for instance, they will regenerate. The axons will regrow, they'll reform the right connections, the frog will recover from paralysis. Those processes don't naturally occur in humans. Christopher Reeve, the guy that played Superman, after his quadriplegic injury falling off a horse, started a whole foundation that's funded some really excellent work in the field of trying to reverse and repair that kind of injury. Doesn't yet exist. Nonetheless, there's a surgeon who has made a name for himself by being rather flamboyant and doing things like this. So this is attaching a rat head onto a rat. Okay, here again, we're talking about vascular supply. So this rat's head had its carotid artery and jugular vein surgically attached into the carotid artery and jugular vein of the host rat here. Did it work? Yeah. Did they get perfusion into the new brain? Yes. Is it functional? No, not outside the functions of the head. So some things are functional. This second rat head still blinks and so on. But does it control anything? No. Perfusion, sure. I'm willing to believe that we are at the stage where we can keep a head alive, basically. You could chop off my head, hook me up to some tubes. Would you be able to maintain my head's function? Yeah, if you get me oxygen and glucose and all the nutrients and so on that I need, I'm willing to believe you can keep my head alive for at least a short time. Can you make my head transplantable and put it on a new body? Even if you can somehow, let's go science fiction here, clone my own body so that I don't have any transplant rejection worries. No, I really don't think so. So Sergio Canavero is this kind of somewhat cranky, possibly a genius, but as far as we can tell right now, crank, Italian surgeon, and he's working with a group in China. He's published a couple of papers in really dodgy journals. This is back in 2016, claiming he'd achieved spinal cord fusion. No data, no evidence, just a report. It seems sadly that this is probably not true. 2017, this is the two rat head piece. It's a real paper, it's a real journal. It definitely happened. Does it mean anything? I'm not so sure it is. And so 2015, the guy we saw in the first slide volunteered. He said, okay, look, what have I got to lose? This works in frogs. Maybe this guy's a genius. My body doesn't work anyway. I'm willing to take the risk. And so there've been several reports, over the ensuing years, it's about to happen. It's about to happen. 2017, nothing happened. And then as you saw, although he's reported, that he's done these head transplants, no data. The first human head transplant carried out on a corpse. Great, wonderful. What exactly does that mean? So do we think this has happened? No, I don't think any humans have had head transplants. The guy that was responsible for the volunteering has now backed out. Apparently with the publicity, made him sufficiently famous that he found a girlfriend, has fallen in love, plans to have a family. So, yeah. Here's a slightly different take on it, okay? So this is an individual, a toddler in fact, whose head was basically knocked off. So what happened was, at the C1, C2 level, here's a normal brain, they cracked their spine. So the spinal cord is still intact, but the spine was broken. And what the surgeons did was, properly align it. They couldn't use screws because they were too small. So the surgeon used wire to reattach the spine and basically hold it together, using baling wire, basically using like paper clips, tiny paper clips. And then because of the wire's limited strength, so what you see here is they actually took one of his ribs, took this toddler's bone from his ribs, and basically used it as a splint for the spine. Is that miraculous? Yeah, it's a wonderful act of surgery, but notice the spinal cord was still intact. So it's not like the head was really reattached. The spine was repaired, but was the actual spinal cord? No. So, can we do this stuff in an adult human? No, I don't think it's so. All right, one other thing I wanted to revisit, and I forgot to talk about this at the time, we'd been talking about drugs of addiction and the fact you can get addicted to all kinds of things, including running. And there was a quote I just loved in this piece from The Guardian about ultramarathon runners. I know a couple of ultramarathoners, those guys are nuts. But they go out and run for literally days on end. Sometimes the longest race my friend Scott has run was 250 miles. And that was a desert race. I mean, the serious endorphins needed. But I love this quote. It makes you feel so good, but after about 4 to 5 hours, you get really sore. So it kind of prevents it from being addictive. That's not true. The addiction is definitely still there, but I can see how it might be limited by also creating this memory association between running and eventual pain. It's going to limit the strength of your addictive memory. You might be able to see, I hope, different bits of the cause. Addiction, memory, pain memories versus addiction memories, how we create this engram of associations. All this stuff does really come together in terms of interpreting real life situations. All right, back to more kind of mainstream class content. We talked a bunch last time about this paper about reconsolidation and the use of anisomycin, the confounds that created with the neurotransmitter storm basically being released in the amygdala. Here's the summary slide again. We went through this in detail. We also ran through a bunch of experiments and a bunch of evidence for different steps in the memory mechanism cascade I've been putting together. Everything from the NMDA channel to calcium coming in to gamma kinase 2 activation, leading to AMPA channel translocation and CREB activation. And so all those data really do hang together. This use of the probe trial. So here's the water maze tank. Here's the target quadrant. Here's where the platform is normally under the water. And you can see that animals that have good memory will spend most of their time exploring that target quadrant, even when the platform's taken away. They know, hey, it should be over here. Animals with impaired memory, for a variety of reasons, don't show that kind of memory. They'll just kind of explore at random. They may have got faster over time, in part because they have a more efficient swimming strategy, perhaps. Rather than sticking to the edge of the tank, maybe they now search systematically throughout the water maze. But they show no evidence of remembering where the platform actually is. When we talked about the differential activation and involvement of NMDA channels only when LTP is happening, when learning is happening, but of the AMPA channels being involved both during learning in order to allow that synapse to get stronger, to depotentiate, to depolarize, and therefore to potentiate, and also during retrieval, during playback of the memory, in order to allow the rat or the mouse to demonstrate they've learned something. The engram that is created by this formation of LTP moves from the hippocampus out to the cortex with a time course of years and decades. This can be a very slow process. And there's a much more advanced course I teach for graduate students about exactly how that engram is organized and whether maybe the hippocampus isn't indexed somehow, which is why you still see some kind of hippocampal involvement in some patients for many years. But certainly in general, the set of neurons involved in storing a particular memory moves from being primarily hippocampal to primarily cortical over the course of some years. Insert the typical question slide here. And I left you with this kind of teaser about, okay, this is how memory is formed. Can we go in and mess with it? Can we take out a memory? And why would we want to do that? I will note, as in passing, that this whole area has been reviewed in 2015 in a really well-written, really accessible paper. Sheena Jocelyn heads a lab up in Canada that's one of the world's leading labs on engram mechanisms and storage mechanisms. And I really like her work. In general, I think she's a smart, really clearly designing scientist who creates experiments that are clearly interpretable with lots and lots of thought about how you might eliminate alternative explanations. So I really like her work. It's a great paper. It's on Blackboard. I really encourage you to go read it. I'm not going to quiz you on it on the final exam, and I realize that means your chance of reading it just went down. But if you're interested in memory at all, it's a really worthwhile paper. All right, so from her lab, the Hahn paper, and this is fair game for tests and so on. I'm hoping that by the time you guys actually get to watch this video, I'll have a class presentation for you on this paper also. What these guys are going to do is create the ability to selectively delete a certain set of neurons. And so here's the methods kind of build up. They've got a mouse that expresses the diphtheria toxin receptor. So normal wild type mice don't have this receptor. Diphtheria toxin doesn't affect wild type mice. You can't kill a wild type mouse with diphtheria because it just doesn't have the receptors for it. That receptor is under control of a stop codon. But notice that stop codon is surrounded by LOX-P sites, right? We've seen this mechanism before. Cre-LOX is a mechanism for selectively snipping out a bit of DNA. So in this mouse, they have the potential to express diphtheria toxin receptors. But in fact, they don't, because just upstream of that in the chromosome is a stop codon. It's a thing that says, no, don't turn on this gene. So the gene is there, but it's never used. All right. If you give an injection of a virus that contains Cre. Cre is the scissors that cuts, the LOX-P. So if you give a vector of a virus that contains Cre, then any cell that has that virus taken up into it will now make some Cre. And what that means is they'll make diphtheria toxin receptors. They'll start making these little cell surface molecules that make them vulnerable to diphtheria toxin. I'm going to walk through that again. Every cell in this mouse has the gene for the diphtheria toxin, but it's not turned on because it has a stop codon that says, don't turn on this gene. If you give an injection, a viral vector that contains Cre, that virus will get taken up by some cells. Whichever cells it gets taken up by will cut out that stop codon, will turn on the gene, and will now be vulnerable to diphtheria toxin. If you then give the mouse an injection, whole body injection of diphtheria toxin, what's going to happen is that only the cells that took up that virus are going to die because they're the only ones that are vulnerable. The cool thing about the vector these guys used is it did two things. It didn't just have Cre, it also contained the gene for CREB. So CREB, you should recall from last class, CREB is this transcription factor active in the nucleus that turns on memory mechanisms. And so what's happening in this particular experiment is that they're using a treatment that does two things. Any cell that takes up this vector will first of all cut out the stop codon because it has Cre, will be vulnerable to diphtheria toxin. It will also make much more CREB. And so the CREB basically makes the neuron poised to form a memory. Their idea was, maybe we can select for which neurons are going to make up the engram for a memory by giving them extra CREB to start with. And so it's as though those neurons are sitting there extra primed, ready to form a memory. If they look to see which cells contain ARC after a memory. So ARC is this early gene associated with plasticity. So expression of ARC suggests that particular neuron has been involved in forming LTP, has been involved in storing some experience. What they showed was that in fact, giving CREB-Cre, giving this viral vector that contained extra CREB, caused an increase in ARC neurons. and it's expressed over here. What they did basically was tag GFP into the same vector so they could see whether they were making more ARC positive neurons in the cells that took up the vector, and the answer is yes. It's a kind of complex readout. I'm not going to go into it again, don't stress it. The overall interpretation is very clear. The interpretation is by giving this vector that had extra CREB, the neurons that took up that vector were much more likely to also express ARC and therefore to be taken as showing plasticity. So having more CREB did seem to make those neurons more likely to express plasticity to have LTP. Here's the key summary slide from the paper. And there are 4 different groups. And the groups are varied in 2 things. They're varied in which vector they got, which virus, and they're varied in whether they got diphtheria toxin or not. So here's the logic. All right. What you're seeing down here is kind of a cartoon of neurons. All the blue neurons are not involved in this experiment. Any purple neuron is a neuron that was involved in the particular memory that's being created. And any white blob is a dead neuron. All right, so take your animals. Give them a vector injection. And the vector is either control with some Cre, so they're getting Cre, but there's no extra CREB anywhere. Or they're getting just CREB by itself, so some cells are getting extra CREB. Or they're getting both. They're getting CREB, Cre in the same vector. So some cells are going to get extra CREB and be vulnerable to diphtheria toxin. So you give them this injection, you wait 3 days. So the virus gets in, it replicates, and so on. You then train them. You make a fear memory that will cause freezing when they hear a particular sound. You test that memory the next day. So you see, do the animals actually freeze? And then you give the animals an injection, either of diphtheria toxin or of saline, PBS, phosphate buffered saline, a vehicle that does nothing. And then you read out, okay, freezing on day 7. So there are 2 days of testing. Green is day 5, black is day 7. So this group over here, didn't get any extra CREB, but they did get some Cre. So some cells took up this viral vector. Because they took up Cre, they've made diphtheria toxin receptors. And so when you give them diphtheria toxin, you kill off some cells. These are the white blobs. But you're killing off random cells. The virus went into random cells. Only by chance would those same cells be used to make a memory. There's nothing about those cells that makes them especially vulnerable. So they have a baseline level of freezing that's pretty low, but it doesn't go away. Day 5 and day 7, the same. Giving diphtheria toxin didn't cause any harm to the memory. Here's the critical group. CREB, Cre in the same vector. So whichever neurons take up this vector are making more CREB, so they're going to be the ones storing the memory. And they are vulnerable to diphtheria toxin because they have Cre. So on day 5, memory got better. They've got extra CREB, so they're better able to store this memory. They've got neurons that are primed, ready to form memory really easily. So they do more freezing. Then you give them diphtheria toxin, and those same cells that had the extra CREB and stored the memory are the same cells that are vulnerable to diphtheria toxin because they also have Cre. So all these little purple dots where the memory was formed have now been wiped out. What you see then on day 7 is this marked impairment of the memory. In fact, freezing is below baseline. And so when the paper talks about selective erasure of a fear memory, it's this group they're talking about. The same neurons that had CREB and therefore were storing the memory were also the ones that were killed off by diphtheria toxin. A couple of control groups. If you give extra CREB but no Cre, then you never make diphtheria toxin receptors. And so giving diphtheria toxin doesn't kill off your memory. But you can see you've got extra CREB, and so your memory is better than your control subjects. And likewise, if you give extra CREB and you give Cre, so you've made these purple cells, but you don't give any diphtheria toxin at all, you just give saline, obviously no cells die off, and so again. So all these groups all have enhanced memory compared to the controls, but it's only the ones where the CREB and the Cre are in the same neurons, and then you give them diphtheria toxin to kill them off, that you see this impairment. Hope that was clear. If not, do ask me questions about it. I think it's pretty, relatively simple logic train to walk through and a clever set of methods. And yes, they definitely did go in and wipe out just that one fear memory. Well, okay, that was a cool thing to do in mice. How about humans? We talked last time a bunch about reconsolidation. So Sheen and Jocelyn, the hand paper, didn't reconsolidate a memory. They just made a memory and then gave a chemical that wiped out the cells containing that memory. That's probably not what you want to do to the human brain. How about if we can just prevent that memory from being restored? So we saw in reconsolidation, you give an easemycin. If you give an easemycin when the animals are being reminded of that particular memory, reminded of the fear memory, the dark space, then you bring back again a day later, that memory appears to have gone away. And so Joe Ledoux and Liz Phelps are two leading neuroscientists at NYU working on, in general, mechanisms of fear memory. And so the approach here is to say, okay, if we remind people of a memory and then we mess with that memory, can we get it to go away? Can we prevent fear from returning? So here's the basic experimental approach. And you're going to see something very similar to this for a bunch of studies. First, make people afraid, create a fear memory. And the way they did this was basically have subjects hooked up to little shock generators. And then on the screen in front of them, they would have colored squares pop up. And in this case, I think it was an orange square, nothing bad happened. And a blue square was associated with a shock. And you can measure all kinds of measures. You can measure pulse rate, you can measure sweating. You can tell whether the subjects are afraid. So day one, all groups go through this process. You create a fear memory in all these groups. Here's the key part. On day two, the groups differ. One group gets no reminder trial. And what that means is they're all going to have extinction, where you repeatedly pair that scary blue square with no shock. So you're basically going to try to extinguish the memory. You're going to try to get rid of the memory that the blue square is scary. One group, there's no reminder trial. The other two groups both get a reminder trial. That is, the first thing that happens is they get a blue square and a foot shock, sorry, finger shock. You basically reactivate the memory, right? You activate the memory from the previous day. Oh yeah, blue squares, scary. And you activate that memory either 10 minutes before extinction training or 6 hours before extinction training. And on day three, you check and basically say, hey, did the memory really get extinguished on day two, or is it still there? Here are the data. Day one, everyone looks like they've formed a memory about the same strength. Don't worry about the y-axis here. Just take it as a measure of fear memory. Day two, after extinction, everyone looks like they've lost the memory. They're no longer responding to a blue square. It's not making them sweat. It's not causing them to get pulse rate to increase. So all the groups look like the memory's gone. Day three, bring them back and see whether the memory has really gone. And the answer is the memory has really gone only for the group that got the reminder trial just before extinction. The other two groups, even though they looked as though the memory had gone away on day two, it's actually still there. Their response is almost as big as it was on day one. So basically what the interpretation is that this group, where the reminder was just before the extinction training, basically that memory was brought back into what's called a labile state, a state where it can be messed with. You've caused it to come up to the front of the brain into kind of short-term memory, and then you've interfered with it. Then you've gone through this extinction training. In these other two groups, you've gone through the extinction training, but the memory wasn't in that vulnerable state. You didn't remind them, you didn't activate the memory just before the extinction. Either you didn't remind them at all, or you reminded them too far advance. The memory wasn't there in a vulnerable state to be reconsolidated. Effectively, what you were doing was maybe trying to create a second separate memory of blue squares not being scary, but it didn't wipe out the original memory. The original memory, in fact, of blue squares being scary is still there, unless you were reminded just before the extinction training. Really cool follow-up. So this is, 1 day, 2 days later. What they did actually was then try to find people a year later. So they actually tracked down 19 of the students who'd originally taken part in this experiment, and then tested them again, showed them a blue square, asked what happens. Here's the guys that had the 10-minute reminder, so they were reminded just before the extinction, still not scared. Here's people from the two groups where they either got a reminder 6 hours in advance or got no reminder, and they're still scared. They're still showing a significant fear response to that little blue square on a computer screen a year later. The memory has been established and has persisted for a year, even though it's, you know, an artificial lab environment of a blue square being a little bit scary. That memory's persisted, but so has the elimination. The group where the memory was activated and then immediately extinguished, that memory's gone. Follow-up experiment. Is it that that one specific memory is being eliminated? Or is this less precise? Because, you know, let's say you have a fear of spiders. We want to wipe out your fear memory for spiders. We don't want to wipe out your fear memory for, oh, some politician that you hate, or a guy pointing a gun at you, or whatever it might be. We only want to wipe out that one particular memory. So here's the trial. 3 stimuli, CS, conditioning stimulus, 2 of which, in this case it was blue squares and green squares, are both associated with little finger shock. So you're learning to be afraid of both of those. And orange squares, not scary. So 3 different stimuli, blue, green, orange, 2 of which are making a fear memory. On day 2, you then remind subjects of 2 of those things. You remind them about blue squares being scary and orange squares being not scary. Then you go through extinction for all 3. So you're extinguishing blue squares and green squares. You're repeatedly putting up blue squares and green squares, neither one of which is having anything scary happening. So you're extinguishing both of those stimuli. And then you're asking, well, which memory actually got eliminated? And as you might expect, right? Here's this freezing or sweating or whatever the index is. Blue squares and green squares, both scary. Orange squares, not scary. After extinction, no squares scary. So far, so good. Day 3, bring them back and say, which is now scary? The one that you were reminded about, blue squares, nope, that fear memory's gone away. It's really not scary, that memory's gone. Green squares, even though you went through the same extinction training, but you didn't get a reminder, green squares still scary. So it's specific. Whichever memories were activated just before the extinction training, those are the memories that have been specifically deleted. So it's actually pretty cool data. You can take a particular fearful memory, and by activating it and then extinguishing it, by then repeatedly saying, no, this memory is not scary, you can minimize or eliminate that fear memory. Think again about, fear of spiders. We activate the fear of spiders, and presumably that's a pre-existing scary memory. So we remind you of that fear of spiders. We actually pair that fear of spiders perhaps with a little finger shock. And then we show you lots and lots and lots of pictures of spiders, and nothing bad happens. Maybe we even feed you jelly beans. So we're making a new memory that, hey, spiders are cute and tasty, and I get candy. Because we immediately reminded you of that memory beforehand, your memory for spiders is in this labile, vulnerable, alterable, malleable state. And so you come back the next day, and spiders aren't scary now. Spiders give you candy. Excellent. And I'm making light of it, but that really is a real thing that can be done. This really is a real approach that is being used to treat different phobias. If you look using MRI, what about the memory trace in terms of metabolic activity? So this is an fMRI study, functional magnetic resonance, looking at amygdala activity. Exactly the same design, exactly the same timing almost. So make a memory, activate either 6 hours ahead or 10 minutes ahead. They stole the exact protocol from the previous experiment. And what they saw was that even on the amygdala activation level, yes, subjects that had a reminder 6 hours before, extinction didn't work, right? The amygdala was still active. Subjects that had a reminder 10 minutes before extinction, absolutely. The amygdala was less active. And you can see here the two different, so these are people that had reminders 6 hours ahead of time, 10 minutes ahead of time. There's just very obviously much less amygdala activity in response to that potentially scary stimulus. Let's take this a step further. If addiction is a memory, and in large part I've suggested that it is, can we use this same approach to go in and wipe out the addiction memory as a tool for preventing relapse, as a tool to aid in the process of breaking a drug addiction? You wouldn't get a paper published in Science if the answer weren't yes. And again, this is very, very similar work. So first of all, they did the kind of proof of principle in rats. Take a bunch of rats, make them go through a conditioned place preference. So this is a task where you make the rat really like a particular environment with a reward. You give them several days of training on this task where the reward is morphine. You're basically taking a whole bunch of rats and getting them hooked on morphine. And yeah, this is ethical and approved. You make sure that you've got that strong preference. The rats are really going to this one particular place. And then again, you get these very similar groups. The 10-minute beforehand group, the 6-hour beforehand group, they added a 1 hour beforehand time point for kicks, and then no reminder at all. Wait a day, test to see what happens. Wait a further day, test again. Data are just what you would expect. Baseline craving for morphine, nada. Craving for morphine as expressed by how much the rats prefer the location where they get the morphine, very strong in all groups. So we're here, all the groups have very strong craving. You can extinguish, apparently, in all groups. So you put them all through extinction where they don't get any reward, and you can make it look as though that memory's gone in all the groups. End of that day, no rat is showing a preference for the place where they used to get morphine. Then you bring back again, and look, animals that got no reminder, so they didn't get a trial paired with morphine to make the memory active on extinction day, or animals that got a reminder but then had to wait 6 hours before extinction. Those two groups still show a very clear memory and desire and preference for the location that has morphine. The other two groups, either 10 minutes or apparently 1 hour before extinction training, the memory is still vulnerable. 1 hour is apparently not long enough to make the memory go back into long-term memory. It's still active. It's still in that vulnerable short-term memory state. And so those two groups really do show no craving. You bring them back. This is, I think 6 days later. You see no retrieval for reinstatement of craving. Basically, this is trying to reactivate and re-hook the rat. And what you see is that the animals that have no retrieval have a much more rapid nose poke response to being re-hooked on the same stimulus. Animals that had gone through the whole reminder, retrieval, extinction protocol, less vulnerable, will make fewer responses given a particular reinstatement attempt to get them back re-hooked. What about humans? You can't take a bunch of undergrads and randomly assign them to get hooked on morphine or heroin. So they had existing, heroin addicts. These are folks who experience heroin cravings. You put them through a very similar protocol. You either show them a 5-minute video about something neutral, Britney Spears or whatever, I really have no idea what they're. And then two other groups, both of whom get a 5-minute video about heroin. So as you might expect, you show them syringes and pictures of people getting high and shooting up. That's going to definitely activate these addicts' cravings for heroin. And then put them all through extinction training. And extinction training here is repeatedly pairing the craving stimuli, pictures of syringes, pictures of places where they used to get high and so on, in this case, with a negative stimulus. I think they actually put them through little shocks linked to that particular craving. And then you look to see, did you eliminate the memory? Did you reduce the cravings in response to a cue that is linked to heroin? 4 days later, a month later, 6 months later, all right? So it's a 1 hour treatment, and we're looking 6 months later. Total cost per patient, maybe $30 at most. And what they saw was that even up to 6 months later, the group that got the reminder just before extinction, which is what the group you'd expect to be effective here, right? Is their cue craving. It's markedly reduced. 6 months looks just like 4 days. The amount of cue induced craving is massively diminished by that one hour intervention. It really does look as though you can take an addictive memory, reactivate it, extinguish it in 1 hour, and markedly reduce the chance in 6 months' time that you're going to reactivate that memory, and presumably the chance that you're going to relapse and go seek out drug. Very impressive data. This approach is absolutely being used. Not so much in the US, because treatment for drug addiction in the US is messed up entirely. But some places in the US and certainly around the world, it's being used very effectively. All right, I told you that we could, eliminate memories. What about putting memories in place? Next slide is a TED Talk. It's actually one of 2 videos I'm going to show you here. This one's from 2012, is a little old. The follow-up is from 2019, is much more recent, but also much vaguer. But this is reporting one of the very first studies to be able to use optogenetics to artificially implant a false memory in the brain of a mouse.
My first year of grad school, I found myself in my bedroom eating lots of Ben & Jerry's, watching some trashy TV, and maybe listening to Taylor Swift. I had just gone through a breakup. So for the longest time, all I would do is recall the memory of this person over and over again, wishing that I could get rid of that gut-wrenching, visceral blah feeling. Now, as it turns out, I'm a neuroscientist, so I knew that the memory of that person and the awful emotional undertones that color in that memory are largely mediated by separate brain systems. And so I thought, what if we could go into the brain and edit out that nauseating feeling, but while keeping the memory of that person intact?
So in fact, this is even slightly more advanced than our previous discussion. Because the emotional aspect of this is largely driven by the amygdala, and the kind of declarative aspect is largely hippocampal. What Steve Ramirez is going to try to do, in theory, is not lose the memory. He still wants to be able to, let's say, remember the person that he used to be in a relationship with. But he wants to eliminate the negative emotional aspect of that memory also, only. So not even just get rid of the memory, which is kind of reasonably simple, just whack someone on the head and they'll forget the last 5 minutes. He wants to be able to go in and alter and mess with this memory to remove just part of it, to remove the negative emotional bit.
And then I realized maybe that's a little bit lofty for now. So what if we could start off by going into the brain and just finding a single memory to begin with? Could we jumpstart that memory back to life? Maybe even play with the contents of that memory. All that said, there is one person in the entire world right now that I really hope is not watching this talk. So there is a catch. These ideas probably remind you of Total Recall, Eternal Sunshine of the Spotless Mind, or of Inception, but the movie stars that we work with are the celebrities of the lab.
Test mice. As neuroscientists, we work in the lab with mice trying to understand how memory works. And today, we hope to convince you that now we're actually able to activate a memory in the brain at the speed of light. To do this, there's only 2 simple steps to follow. First, you find and label a memory in the brain, and then you activate it with a switch. As simple as that.
Are you convinced?
No.
So turns out finding a memory in the brain isn't all that easy.
Indeed, this is way more difficult than, let's say, finding a needle in a haystack. Because at least, the needle is still something you can physically put your fingers on, but memory is not. And also, there's way more cells in your brain than the number of straws in a typical haystack. So yeah, this task does seem to be daunting, but luckily we got help from the brain itself. It turned out that all we need to do is basically to let the brain form a memory, and then the brain will tell us which cells are involved in that particular memory.
So what was going on in my brain while I was recalling the memory of an ex? If you were to just completely ignore human ethics for a second and slice up my brain right now, you would see that there was an amazing number of brain regions that were active while recalling that memory. Now, one brain region that would be robustly active in particular is called the hippocampus, which for decades has been implicated in processing the kinds of memories that we hold near and dear, which also makes it an ideal target to go into and to try to find and maybe reactivate a memory.
When you zoom in into the hippocampus, of course, you will see lots of cells, but we are able to find which cells are involved in a particular memory, because whenever a cell is active, like when it's forming a memory, it will also leave a footprint that will later allow us to know these cells are recently active.
So the same way that building lights at night let you know that somebody is probably working there at any given moment, in a very real sense, there are biological sensors within a cell that are turned on only when that cell was just working. They're sort of biological windows that light up to let us know that that cell was just active.
So we clipped part of this sensor and attached that to a switch to control the cells. And we pack this switch into an engineered virus and inject that into the brain of the mice.
This is a very similar approach to we've been discussing with previously. This is a viral vector. Often we use an adenovirus, which is the common cold, or herpes-associated virus, or modified rabies virus. All these are very effective at infecting cells. Obviously, you take out all the disease stuff first and replace that payload, that content, with the genes that you want to express. In this case, it's going to be a rhodopsin channel, so a channel that will make that cell responsive to light.
So whenever a memory is being formed, any active cells for that memory will also have this switch installed.
So here's what the hippocampus looks like after forming a fear memory, for example. The sea of blue that you see here are densely packed brain cells, but the green brain cells are the ones that are holding on to a specific fear memory. So you're looking at the crystallization of the fleeting formation of fear. You're actually looking at the cross-section of a memory, right?
And think about how many cells that was. So this is a simpler experiment where they're simply using a green GFP, green fluorescent protein, to mark all the cells that are active. That's a lot of cells. So the hippocampus uses a whole bunch of neurons, even for a very simple memory. The exact pattern varies depending on what they're forming memory of, and it's going to link to different sets of sensory neurons. So if it's a memory to be afraid of a toad, it'll link to auditory neurons, which will also be active to make the whole engram. Well, that's a ton of neurons being involved in even a very simple memory.
Now, for the switch we've been talking about, ideally, that switch has to act really fast. It shouldn't take minutes or hours to work. It should act at the speed of the brain in milliseconds.
So what do you think, Shu? Could we use, let's say, pharmacological drugs to activate or inactivate brain cells?
No, drugs are pretty messy. They spread everywhere. And also it takes them forever to act on cells. So it will not allow us to control a memory in real time. So Steve, how about let's zap the brain with electricity?
So electricity is pretty fast, but we probably wouldn't be able to target it to just the specific cells that hold on to a memory, and we'd probably fry the brain.
Oh, that's true. So looks like we need to find a better way to impact the brain at the speed of light.
So it just so happens that light travels at the speed of light.
A little corny.
So
yeah, okay,
maybe we could activate or inactivate memories by just using light.
That's pretty fast.
And because normally brain cells don't respond to pulses of light. So those that would respond to pulses of light are those that contain a light-sensitive switch. Now to do that first, we need to trick brain cells to respond to laser beams.
Yep, you heard it right. We are trying to shoot lasers into the brain.
And the technique that lets us do that is optogenetics. Optogenetics gave us this light switch that we can use to turn brain cells on or off. And the name of that switch is channel rhodopsin, seen here as these green dots attached to this brain cell. You can think of channel rhodopsin as a sort of light-sensitive switch that can be artificially installed in brain cells, so that now we can use that switch to activate or inactivate the brain cells simply by clicking it. And in this case, we click it on with pulses of light.
So we attach this light-sensitive switch of channel rhodopsin to the sensor we've been talking about and inject this into the brain. So whenever a memory is being formed, any active cell for that particular memory will also have this light-sensitive switch installed so that we can control these cells by the flipping of laser, just like this one you see.
So let's put all of this to the test now. what we can do is we can take our mice and then we can put them in a box that looks exactly like this box here. Then we can give them a very mild foot shock so that they form a fear memory of this box. They learn that something bad happened here. Now with our system, the cells that are active in the hippocampus, in the making of this memory, only those cells will now contain channel rhodopsin.
When you are as small as a mouse, it feels as if the whole world is trying to get you. So your best response of defense is trying to be undetected. Whenever a mouse is in fear, it will show this very typical behavior by staying at one corner of the box, trying to not move any part of its body. And this posture is called freezing. So if a mouse remembers that something bad happened in this box, and when we put them back into the same box, it will basically show freezing because it doesn't want to be detected by any potential threats in this box.
So you can think of freezing as you're walking down the street minding your own business, and then out of nowhere, you almost run into an ex-girlfriend or ex-boyfriend. And now those terrifying 2 seconds where you start thinking, what do I do? Do I say hi? Do I shake their hand? Do I turn around and run away? Do I sit here and pretend like I don't exist? Those kind of fleeting thoughts that physically incapacitate you, to temporarily give you that deer in headlights look.
However, if you put the mouse in a completely different new box, like the next one, it will not be afraid of this box because there's no reason that it will be afraid of this new environment. But what if we put the mouse in this new box, but at the same time, we activate the fear memory using laser, just like we did before? Are we going to bring back the fear memory for the first box into this completely new environment?
All right, and here's the $1 million experiment. Now, to bring back to life the memory of that day, I remember that the Red Sox had just won. It was a green spring day, perfect for going up and down the river, and then maybe going to the North End to get some cannolis, hashtag just saying.
Not to interrupt, but think about all those different aspects that are all going to be part of the whole memory. You don't want to wipe out, ideally, your enjoyment of the spring day, the happy bird noises, the river, and so on. You just want to get rid of the fact that you're about to get mugged, right? Wipe out that little bit of the memory in a particular example.
Now, Shu and I, on the other hand, were in a completely windowless black room, not making any ocular movement that even remotely resembles an eye blink, because our eyes were fixed onto a computer screen. We were looking at this mouse here, trying to activate a memory for the first time using our technique.
And this is what we saw. When we first put the mouse into this box, it's exploring, sniffing around, walking around, minding its own business. Because actually by nature, mice are pretty curious animals. They want to know what's going on in this new box. It's interesting. But the moment we turn on laser, like you see now, all of a sudden, the mouse entered this freezing mode. It stayed at here and try not to move any part of its body. Clearly, it's freezing. So indeed, looks like we're able to bring back the fear memory for the first box in this completely new environment. While watching this, Steve and I are as shocked as the mouse itself. So after the experiment, the two of us just left the room without saying anything. After kind of long, awkward period of time, Steve broke the silence.
Did that just work?
Yes, I said, indeed it worked. We're really excited about this. And then we published our finding in the Journal of Nature. Ever since the publication of our work, we've been receiving numerous comments from all over the internet. Maybe we can take a look at some of those.
So the first thing that you'll notice is that people have really strong opinions about this kind of work. Now, I happen to completely agree with the optimism of this first quote because on a scale of 0 to Morgan Freeman's voice, it happens to be one of the most evocative accolades that I've heard come our way. But as you'll see, it's not the only opinion that's out there.
Indeed, if we take a look at the second one, I think we can all agree that it's probably not as positive. But this also reminds us that although we are still working with mice, it's probably a good idea to start thinking and discussing about the possible ethical ramifications of memory control.
Now, in the spirit of the third quote, we want to tell you about a recent project that we've been working on in lab that we've called Project Inception. So,
we'll talk about movies, don't worry.
Now that we can reactivate a memory, what if we do so, but then begin to tinker with that memory? Could we possibly even turn it into a false memory?
So our memory is sophisticated and dynamic. But if just for simplicity, let's imagine memory as a movie clip. So far, what we told you is basically we can control this play button of the clip so that we can play this video clip anytime, anywhere. But is there a possibility that we can actually get inside the brain and edit this movie clip so that we can make it different from the original? Yes, we can. Turns out that all we need to do is basically reactivate a memory using laser, just like we did before. But at the same time, if we present new information and allow this new information to incorporate into this old memory, this will change the memory. It's sort of like making a remix tape.
So, how do we do this? Rather than finding a fear memory in the brain, we can start by taking our animals, and let's say we put them in a blue box, like this blue box here, and we find the brain cells that represent that blue box, and we trick them to respond to pulses of light, exactly like we had said before.
All right. If you kind of dozed off at any point, this is the cool part.
Now, the next day, we can take our animals and place them in a red box that they've never experienced before. We can shoot light into the brain to reactivate the memory of the blue box. So what would happen here if while the animal's recalling the memory of the blue box, we gave it a couple of mild foot shocks? So here, we're trying to artificially make an association between the memory of the blue box and the foot shocks themselves. We're just trying to connect the two. So to test if we had done so, we could take our animals once again and place them back in the blue box. Again, we had just reactivated the memory of the blue box while the animal got a couple of mild foot shocks, and now the animal suddenly freezes. It's as though it's recalling being mildly shocked in this environment, even though that never actually happened. So it formed a false memory because it's falsely fearing an environment where, technically speaking, nothing bad actually happened to it.
So far, we're only talking about this light-controlled on switch. In fact, we also have a light-controlled off switch. And it's very easy to imagine that by installing this light-controlled off switch, we can also turn off a memory anytime, anywhere. So everything we've been talking today is based on this philosophically charged principle of neuroscience, that the mind, with its all seemingly mysterious properties, is actually made of physical stuff that we can tinker with.
And for me personally, I see a world where we can reactivate any kind of memory that we like. I also see a world where we can erase unwanted memories. Now, I even see a world where editing memories is something of a reality because we're living in a time where it's possible to pluck questions from the tree of science fiction and to ground them in experimental reality.
Nowadays, people in the lab and the people in other groups all over the world are using similar methods to activate or edit memories, whether that's old or new, positive or negative, all sorts of memories so that we can understand how memory works.
For example, one group in our lab was able to find the brain cells that make up a fear memory and converted them into a pleasurable memory, just like that. That's exactly what I mean about editing these kinds of processes. Now, one dude in lab was even able to reactivate memories of female mice in male mice, which rumor has it is a pleasurable experience.
Indeed. We are living in a very exciting moment where science doesn't have any arbitrary speed limits, but is only bound by our own imagination.
And finally, what do we make of all this? How do we push this technology forward? These are the questions that should not remain just inside of the lab. And so one goal of today's talk was to bring everybody up to speed with the kind of stuff that's possible in modern neuroscience. But now, just as importantly, to actively engage everybody in this conversation. So let's think together as a team about what this all means and where we can and should go from here. Because Shu and I think we all have some really big decisions ahead of us. Thank you.
If you're on Twitter, Steve Ramirez is pretty active on Twitter. And if you're not bored of other videos, I'm sure you want more of my voice. No, okay. So I'm going to show you a second video from him. Although looking at my slide set, it appears to have vanished. All right, I'll make sure I put it in next time. Basically an update video saying, hey, here's 2019, here's what's happening now. What's actually up next in my slide set is cute pictures of children. Okay, so this is my younger son. He's 11. You guys have probably seen him come in at some point. Why is there a cute picture of him as a baby on the screen? Because when we're talking about memory manipulation and amnesia and forgetting and so on, there's a really interesting case of amnesia, which is infantile amnesia. People talk about having traumatic birth experiences. That's bollocks. It is not possible for the infant brain to form memories. It's especially not possible for anybody to form a brain, or for anybody to form a memory of the time when they were being born. And there's a reason for that, right? You know, you come out into the world, it's cold, it's, you know, kind of annoying. It was nice and warm and comfortable. Why is this happening to you? You scream a lot. And then you grow up and things are much happier. This is him about 4 years ago now. Yes, he's a cute kid. Okay, fine. So this is him 2 years ago. I still coach his soccer team. I still call it football. And I still tell the kids to tackle each other, which they think means jump on each other as opposed to take the ball away. All right, digression into cute kid pictures aside. What happens when you're being born? It turns out that your brain is turned off. There's this really cool case of infantile amnesia, of this, not just for the first couple of years of life, where it's hard to form specific memories. Think about your first memory of your mother's face. All those memories kind of blur together. And I talked about the fact that, particularly because you have more NR2B units and you have more plasticity, it's actually quite hard to form distinct individual memories because memories keep getting overwritten and modified, and things like your mother's face. You activate that memory a lot, you alter it a lot. It's really hard to have a memory for a specific single instant when you remember. So individual memories from early childhood, pretty rare. Most of you probably have an idea of the first distinct declarative memory you have of a particular moment. It's probably not until you're 3, 4, 5, 6, some age that is an infant. Even more particularly though, when we talk about infantile amnesia, we're talking about the moment or the day of birth, pretty much. And the brain is turned off. There's a really cool reason for that. So first thing to realize is that in the fetal brain, there is little or no glutamate. The major neurotransmitter is GABA, and the brain is actually wired up in this wacky way where GABA is excitatory. So in order to turn the brain off, you've got to turn off GABA signaling. This slide just shows you the response to GABA in terms of membrane voltage. And what you can see is that, you know, in the womb, so this is, you know, pre-birth, embryonic day, so on, GABA is excitatory. It causes depolarization. Obviously, in the adult brain, it causes hyperpolarization. It's inhibitory. But in the fetal brain, and for several days post-birth, postnatal day, P5, and so on is out here. This is a rat brain, by the way. You know, embryonic day 21 is birth. Humans don't give birth after 21 days. You know that. So GABA is excitatory. If you turn off GABA, then the brain will basically shut down because there's no glutamate. The major excitatory transmitter is GABA. If you turn off GABA, everything goes away. What you can see is that right around birth, so P0, day of birth, GABA becomes temporarily inhibitory. So the brain really does turn off. There's nothing, you know, causing brain excitation. In particular, there's no plasticity. There's no memory formation going on. There is no, you know, major excitatory transmitter because GABA is being turned off. Then it comes back after birth, you know, by P1, by the end of the first day of life, it's now excitatory again, the brain's back in function. The two red symbols here, those are the consequences of inhibiting oxytocin. So you guys probably have heard of oxytocin as like the trust hormone or something. It's also responsible for forming a very strong emotional attachment between the mother and the child during birth. If you block oxytocin, the brain never turns off. And so there's a whole bunch of complex mechanisms here. Why you'd be getting this drug, excellent question. But it turns out oxytocin is responsible for triggering this movement of GABA from being excitatory to being inhibitory. This shows you the same thing without treatment. So here's birth. Here's the potential change for, you know, a GABA neuron. It's the membrane voltage. And what you can see is they're actually pretty excitable most of the time, but they also turn off right around the age of day of birth. So the neurons are much less active, and when they are active, they're inhibitory. If you turn off oxytocin, all that stops. Oxytocin, it turns out, is released by the pressure of the fetus on the birth canal. So it's a maternal oxytocin release triggered by the whole process of giving birth. Every time I give this particular bit of a talk, I'm asked, what happens if you have a C-section? Well, I was born by C-section. And yeah, in fact, in theory, right, I could potentially have temporarily had a memory of the act of being taken out of the womb. My brain wasn't turned off in the same way. Nonetheless, after a few days at most, that would be overwritten by all kinds of other memories. So no, you still can't have a memory of being born, even if you don't have a C-section. You may have a slightly impaired initial bonding with your mother because you don't get that initial oxytocin release. Separate question. All right. Why is the brain turned off at this time? Think about the process of being born, particularly in humans who are really born kind of 2 or 3 months too early. The only reason the human baby is delivered when it is because at that point it's as big as possible to fit through the pelvis. The human baby still isn't able to survive by itself compared to most other mammals. It's really pretty incompetent. But nonetheless, evolution says you've got to get out now because otherwise you can't get out. You won't fit. And so the brain keeps swelling, the head keeps growing after birth. The process of being born, you're being squashed into this tiny little space. You know, your placenta and umbilical cord, where all your fuel supply are also being compressed. And if you're a really stupid fetus, again, like me, this is why I had a C-section, you've managed to, in fact, strangle yourself with your own umbilical cord so that your blood oxygen supply is going to your brain almost zero. Why do we turn off these GABA neurons? Why do we turn off the brain? Evolutionary, the pressure is so that they can survive for longer when fuel supply, when oxygen supply is interrupted. Because you are being pushed out into the world at this time, basically of great danger, a great risk of interruption to brain oxygen and fuel supply. Turning everything off means the brain needs less fuel, and so the neurons can survive better in a fuel-deprived state. If you look to see how long does it take before the cell just falls apart, baseline conditions, okay, so a normal, fetal hippocampal E21 brain, 55 minutes maybe, they can survive anoxia. If you don't have that oxytocin release, if you block the oxytocin release, it's down to 45 minutes. Does that sound like a big difference? Maybe not, but it's a critical difference in terms of not having brain damage during a period of prolonged anoxic deprivation, which may potentially occur as part of giving birth. So there's this really cool phenomenon of infantile amnesia caused by the whole process of giving birth as a protective mechanism against damage to the hippocampus through anoxia. Speaking of amnesia, if you don't know what this movie this is from, first of all, I'm glad for you. And second of all, we'll talk about it next time. So this is, again, an hour or so of me talking, at least with a little video this time. What we're going to talk about next time, to start with, is some of the movie portrayals of amnesia. Because I mentioned, I think, 2 classes ago, you should watch Finding Nemo. Disney's treatment of amnesia in Dory is actually pretty good. In contrast, Drew Barrymore and amnesia, nowhere near as good. In fact, really quite bad. So we'll have a brief diversion into that. And then what we're going to talk about is some more of the modulation of memory. So, okay, we've talked about how we make memories and mechanisms involved in making memory. You now have a pretty good understanding of NMDA channels and calcium and so on. How do we select which memories? We talked about this in terms of attention. What about on the molecular level? What is going on to cause us to form particular memories for certain stressful events, for instance? You might remember, if you've been mugged, the fear in your experience, the shape of the club someone was using to beat you, those are probably long-lasting, very impactful memories. In contrast, try to think about what you had for breakfast 3 days ago. Much harder, probably made less of an impact on you, unless it was something particularly special. So we'll also talk about modulation and choice of memories, how your brain picks which things to remember. Thanks. I'm going to go make some more coffee. I will talk to you tomorrow. I'll talk to you again on Thursday. Go to the discussion board. Look forward to seeing you there. See you guys.